All right. Last week. Woo. Okay.
Hoorah.
I'm Nathan, and I'll be doing number one.
The glassiest of eyes.
So, number 1A, we're looking at the annealing of a primer and Sanger sequencing. And so the first question is, what would the Sanger sequencing look like? So you have your primer annealing site, and then as a result of it, you've got 4 different wells. Each will have their thing. And so as you go down, this will be the first nucleotide going up to the last nucleotide here, because the first one will be the shortest, so it'll move further down the electrophoresis gel. And then for parts B through D, we're talking about if PCR would fail or still work, depending on the conditions given. So for B, the condition is if we used E. coli DNA polymerase instead of Taq polymerase, and the reaction there would fail because after the first heating up or separating of the strands part of PCR, the DNA polymerase would denature, whereas Taq polymerase is a specific enzyme that is used by bacteria that live in hot pools, so they can survive those hot temperatures. So the reaction would fail because DNA polymerase would no longer work, but Taq polymerase would, which is why we use Taq. And then for C, the addition of helicase. So helicase doesn't really do anything in PCR, so it wouldn't have an effect on the reaction, and so the reaction would still work there. And then for the last one, we add DDATP, but we forgot to add the DDGTP, TTP, and CTP. So all of those dideoxy would not be added, so the reaction would still work, but it wouldn't work how we need it to for the Sanger sequencing, so the reaction would fail overall. The only lane that you would actually have that had multiple bands would be the A lane for the DDATP. And then all of the other ones would just be replicated DNA strands there. And then number 2.
So I got the fill in the blank. So during a typical PCR cycle, the elongation step is done at 72 degrees centigrade, while the annealing step is done at 55 degrees centigrade. AMP-R is a common selectable marker that is found in many plasmid vectors, while restriction enzymes recognize and cleave DNA molecules as palindromic sites. During agarose gel electrophoresis, DNA is placed in an electrical field. DNA migrates towards the positive electrode during electrophoresis. DDATP lacks the 3' OH group and the 2' OH group as well. Transformation is a term used to describe the process where bacteria take up DNA molecules from the environment. During a blue-white screening, IPTG is commonly used to induce the expression of lac operon, while X-gal is cleaved by B-galactosidase to turn E. coli cells into cells of blue. Now, 3. You are interested in using PCR to amplify blah, blah, blah. Here's the region. Write a single-stranded DNA sequence that you would use for your 2 primers in the PCR experiment. So the first one would be a GAGCCTTCACTCAGTC, and that's 5 to 3. And then 5 to 3 again would be CAGTGCAATTGAGGCT. A mathematical equation, blah, blah, blah. Calculate the annealing temperatures of your primers. So if you, all you're doing here is you're counting the C's and G's, dividing by the totals, which is 16, and then just filling out the equation. So the one that started with GAG would be 62.3 degrees centigrade. Depending on how you round, there's a variation of about 0.1. And 59.8 for the CAG sequence. Again, there's about a 0.1 difference depending on how you round. Each PCR cycle contains a DNA synthesis step. What would happen if you 2 newly synthesized strands, if DNT-CTP is accidentally used in the PCR experiment? That would stop the reaction once that C had been implemented in this strand. So you'd have a triple G, A double T, A G, C, because it would stop at that C, and then T, 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 A, A, T, TC, and both of those being in the 3 to 5 direction. And then the reason why we don't use DNA-A or DAM, well, we don't really need it in this reaction due to the fact of the presence of heat. The heat will cause the separation, and so we do not require DNA or DAM to do so. You're up.
You happy to be over for this semester?
I've got 4.
Oh, he's got 4.
Oh, you've got 4.
Rejected.
Changing it up last week. Gotta throw more. Okay, so for this one, we're looking at restriction enzymes digesting a DNA molecule. And so the 3 enzymes we have are BAMH1, EcoR1, HIND3, and then a combination of EcoR1, BAMH1, and then HIND3 and BAMH1, HIND3 and EcoR1. And so the first part is to draw a diagram. Over here we've got the marker lane, and the marker has the ladders of 1,000, 500, 400, 300, 200, and 100. And then we just have the table up here that shows us where we can find those markers on the electrophoresis if we were to run it, or at least how it should look, depending on how good you are at PCR. And so that's how that looks. And then using that information, we'll be able to create this diagram down here, which is a linear chromosome. And then from that linear chromosome, we can tell where the enzymes are and how many base pairs are between them, for a total of 900 base pairs. And then for part C, it's just asking if we were to cleave those molecules, where would all 3 be, or how many base pair fragments we would have if we used all 3 enzymes at the same time. And we'd have 50, 150, 100, 100, and 500 for a total of 900 base pairs. And now we will go on to Josh for number 5.
That was close. That was close.
Almost messed up the video on the last day. Woo! Live it on the edge.
Let's go.
I hope she doesn't grade these too aggressively.
We are sorry for that.
We're never sorry.
But I did. Jacob's never sorry. So the 3 process we had to explain are mRNA to complementary DNA cloning and blue and white screening. So basically, for the first one, we purify mRNA, then we mix with the primer poly-DT, and then we add DNTPs for energy and reverse transcriptase to synthesize a DNA strand. Then we add RNase H to degrade the rest of the mRNA and produce RNA primers. Then we add DNA poly-1 to synthesize the second DNA strand. And kick and keep swimming and cloning, and then we clone the complementary DNA into the vector, and we do that with 5 steps. So we add restriction endonuclease, which cleaves the restriction enzyme site on big beta globulin gene and the vector plasmid. I'm sorry. And then we let the sticky ends hydrogen bond, and then we add DNA ligase to covalently bind sugar phosphate together. And then we insert this molecule into the host cell to be replicated, and then you grow it in the presence of ampicillin. And then hopefully you can watch this with getting it all. And blue and white screening, we do a mix of recombinant DNA with competent E. coli cells, and then we place these on the media. And then
Hold it together, John.
hold it together. And then we observe our results. So the white colonies are vectors with an insert, and the blue are without. All right, and for CRISPR, we have this nice video for you. Basically, Cas9 is a protein that binds with mRNA and makes a complex called CRISPR. And basically, it's an endonuclease that can cleave the DNA, and it can take out viruses.
Simply, it's scientific scissors. And we have a cool little song that kind of describes it in a great way. You might have heard this person or not. We'll just give you the first couple seconds. CRISPR-Cas9, bring me a gene, encoding for a specific protein. Make up your steps at this loaded locus. You work so well inside a snap, go get Cas9. I'm so alone, without your scissors and my chromosome. Cut me up and do it clear. CRISPR-Cas9, bring me a gene. And that's all for this week. I hope you kind of understood us. We were definitely probably not as serious as we normally are.
You got it.
Life.